name's Naomi, and I graduated in 2017 in the biochem department. And I'm at Washington University in St. Louis now, which is in Missouri. And I'm a fourth year PhD candidate in the cow lab, where we study gut and asthma. Oops. There we go. Okay, so first of all, just a little background of asthma. So asthma is a chronic allergic disease, and it's caused by a sensitization to environmental allergens. And this just means that the immune system gets primed to overreact to allergens in the environment, such that when the person is exposed to the allergen again after sensitization, their immune system goes into overdrive, sending immune cells into the lungs and constricting the smooth muscle in the airway, and also telling mucus producing cells in the airway to produce a ton of mucus as a response, a defense mechanism against that allergen that they have been exposed to. So the immune system is doing its job here, but it's just overreacting quite a bit and actually causing damage. And then obviously, like when this happens, the asthma symptoms happen where there's wheezing, airway obstruction, and shortness of breath. So the reason that the immune system overreacts is still kind of a mystery that we're trying to solve. But one of the things we do definitely know about asthma is that the environment plays a very big role. It primes the immune system, it affects asthma risk, and it can cause exacerbations of asthma. And so people have tried to link asthma to genetics in some way, because we do know that asthma is hereditary. And so a lot of targets have been associated with asthma, but we haven't been able to fully explain asthma severity and risk with genetics. And the gut has recently emerged as a mediator of this interaction between the gut and the lung, which kind of makes sense because in the body, it's one of the largest interfaces between the environment and the immune system. And recent studies have directly linked different microbial communities to protection against asthma, different microbial metabolites too, as a mediator of the immune response in asthma. So in our lab, we finished a clinical study that is asking the question in kind of this arena where how can we explain the connection between the gut microbiota and the lung, kind of like what fills in this arrow here. And so in St. Louis, we collected fecal samples from people that have moderate to severe asthma and also just healthy controlled people. And so how do we get from people's poop samples to studying asthma, you would probably ask. So one of the tools we use is germ-free mice. This is something I'd never heard of before I came here, so let's give you a little bit of background. Germ-free mice have, as their name implies, never seen a microbe in their life. They were born in germ-free environments. They're fed autoclaved food and water, and then they also breed in their kind of bubbles. And so here's some pictures of our facility actually. So the mice literally live in these hermetically sealed plastic bubbles that we sterilize, and then we can manipulate them through these gloves in the bubbles. And these are my lab mates here. Okay, so one of the ways that we use these mice and that they're really useful is we can take directly the human fecal samples and use them to colonize these mice so that the only microbes these mice have ever seen are actually an intact human community, and then we can study the effect of that gut community on the mouse phenotype. Okay, so we also have an asthma model. So first, like I said, we colonize these mice with the patient microbiota, and then there's a sensitization and challenge with an allergen that has been used broadly for asthma studies. It's called ovalbumin. But essentially, we inject the mice with a dose of this allergen. The immune system gets primed to overreact to that. And then right before we euthanize the mice and take samples from them, we expose them to this allergen again so that they have the allergic asthmatic reaction. Okay, so one of the things we collect, unsurprisingly, is the lung tissue. And so one of the experiments we do on this is flow cytometry, which is a really powerful tool in immunology to just quantify the number of types of cells, types of immune compartments in this case, the lung. And so on the left here, these are a bunch of markers of T effector cells, which are the immune cells that are going to respond to the allergens in the lung. And we found that those were increased in the mice that received the asthmatic microbiota. And then next is just a subset of those T effector cells that are specifically T cells that produce IL-17. And these comprise this Th17 response, which is kind of a well-known type of asthma. And we saw that those cells were also increased in the mice that received the asthmatic microbiota. And then lastly over here, we confirmed this with just measuring the gene expression in the lungs and found that this IL-17 cytokine was also highly expressed in the mice that received the asthmatic microbiota. And then these other ones are actually from a Th2 response, and those were not different between the mice. Okay, so secondly, we ran RNA-seq on these lung samples, and this time we included a couple control groups that did receive the human microbiota samples, but did not get any asthma disease induced. So they did not receive the sensitization and challenge. Okay, and then on the right is the result of that, where this is kind of a high-level view of the RNA-seq data, where we take a big multidimensional data set and kind of smush it into two axes. It's called a principal components analysis, and basically things that are closer together are more similar, things that are farther apart are more dissimilar. So this clustering tells us that these control groups that did not receive the asthma model were not very different from each other. But then these pink and purple were the ones that did receive the asthma and the microbiota were different. So that's really interesting because it tells us that it's only in the context of the asthma disease that the lung transcriptome is changing. Okay, so next, since we have an immune phenotype and the lung transcriptome is changing just based on the microbiota, we wanted to know what is maybe causing this and what is connecting the two tissues. And so one of our hypotheses was that maybe the gut is being really leaky, and so maybe microbial products are reaching the lung through the circulation just by like escaping the gut lumen. So we measured that directly with this leakage assay, and we found that the mice that did receive the asthmatic microbiota had more gut permeability. And then on the right here, we also measured this through just relative expression of tight junction proteins, in this case occludin, and these are proteins that kind of make the seal tighter in the gut. And so the fact that we saw lower expression of those seal proteins also supports that there is kind of leakage happening in the gut. So next we asked, what are the individuals in the microbial communities that are contributing to this gut permeability and then thus the immune phenotype? And so this is kind of a complex assay as well, but it's basically telling us what bacteria in the gut are the immune is the immune system targeting as like dangerous or pathogenic. And so this big red bubble here tells us that the immune system saying, hey, this is potentially bad for the body. And so it's highly targeted by these antibodies through the immune system. So one of these that came up as highly red in the asthmatic mice was this Bacteroides fragilis strain. And so when we looked further into that, after some whole genome sequencing on that one isolate, we found that it encodes for a toxin called fragilissin. And this is a protease that actually targets tight junction proteins in the intestine and causes diarrhea. So we already know, like through the literature, that this is implicated in gut barrier permeability, which was really cool. But we found this in one sample in our cohort. And so we went back to all of the Mars people and used a targeted PCR to see if we could find this same gene in all of the other fecal samples. And so down here is a table of those results. And with a Fisher's exact test, we were able to say that fragilicin is more present in the asthmatic cohort. And also of the people that had B. fragilis of any type of strain, there were more asthmatics that had the enterotoxic Bacteroides fragilis compared to the non-toxic producing B. frag. So next in this project is to ask what other bacterial species could be playing a role in this gut permeability phenotype we're seeing. And then also, importantly, I'm interested in looking at what other functions are encoded in the gut microbiome that could be playing a role. So taking a more genomics-based approach is probably where I'm going to go with my thesis. So just want to thank my lab, the Cal Lab, especially my co-author, Ariel, and my boss, Andy. And then lastly, this is my contact information. I'm happy to meet with anyone one-on-one on Zoom or whatever you want. Yeah, don't be shy to ask about grad school if that's what you want to do.